Hello all, welcome back to our lectures today. Today we'll be talking about the chemical and physical properties of aldehydes and ketone. Now these chemical properties of aldehydes and ketone will be a two-part lecture. So we'll be looking at the first part of the lecture today. That is why you have where I put it chemical properties 1. So we're going to be talking about the first part of the lecture in this video. All right, let's take it off from there immediately. Now, it's always customary for me to give you a quote and then talk about it. So today's quote is from a wonderful pioneer of the blessed memory, Katherine Johnson. Let's hear what she said. Girls are capable of doing everything men are capable of doing. Sometimes they have more imaginations than men. This was the case of Katherine. Katherine Johnson was born in 1918 and just died recently in 2020. She was a NASA mathematician who was critical to the first US crew in space. She was very crucial to the landing of Apollo 11 on the moon and then back. She worked in time of a lot of discriminations against African Americans. She weathered the storm and showed the potential she has as a mathematician. In fact, she was nicknamed human computer. As at then, we don't have an extensive computer, but she was called a human computer. So in 2015, the first black president awarded the highest civilian honor to her, which is the Presidential Medal of Freedom. So Katherine Johnson was a wonderful and very astute STEM mathematician and a NASA operative until she retired in 1986. Alright, so let's take it off from there immediately. Now, the two learning objectives we'll be covering in this particular lecture is number one, we're going to write or draw aldehydes and ketone oxidation reaction. We're going to look at the oxidation reaction of both aldehydes and ketone. And two, we're going to also write or draw the aldehydes and ketone reduction reaction. Now, in the second part of this chemical reaction lecture, we'll be talking about the hemi formation, but that is not covered in this lecture. Let's see. Oxidation reaction. Now, it's important we know this. Remember, these reactions are usually what you use in doing qualitative analysis or trying to separate a class of compound to another. So what is the difference between the aldehydes and ketone oxidation reaction? Now, aldehydes are oxidized to carboxylic acid by a variety of oxidizing agents such as potassium permanganate or potassium permanganate or potassium dichromate. Now, look at the difference. I'm going to try to use a different color. Ketones do not undergo oxidation reaction. So if I ask you what's the chemical difference between aldehydes and ketone, what you're going to give me is that whereas aldehydes undergo oxidation reactions, ketones do not undergo oxidation reaction. What is the reason? Because ketones are already oxidized. Let's see example. Now this is an aldehyde. Remember, an aldehyde is known by having the carbon, and a hydrogen. If this undergoes oxidation here, remember there are two ways to define oxidation. Oxidation is either the addition of oxygen or removal of hydrogen. Remember that. So in this case, what is happening is that one atom of oxygen is being inserted at this point. And when you insert it there, what happens? It forms carboxylic acid. So the oxidation of aldehyde forms carboxylic acid, whereas ketones do not undergo oxidation. If you use, no matter how strong the oxidizing agent is, if you bring it very close, no reaction occurs at this point. And what is the reason? The reason is because this is already highly oxidized. There is no hydrogen there for it to fit in the oxygen. So the ketone is already completely oxidized. It's completely oxidized and cannot undergo any further oxidation. Now let's take examples immediately. It says complete the oxidation reaction below. Let's try to complete these oxidation reactions. Now, taking a look at this, what do you see? You find out that this is an aldehyde, so eventually it is going to undergo oxidation reaction. Let's see what's going to be happening at this point. Now, what will be happening? You see, okay, let me try to maintain the same color that I used. I wanted to change. It's okay. Now, this oxygen, like I told you, will now be added at this point. So what you basically need to do is to redraw this molecule towards this side and put an O in between this hydrogen and oxygen, and it becomes carboxylic acid. So if I do that, I'm going to start with what I have here. I'm going to have this, I'm going to have this, I'm going to have the carbonyl carbon here, and then I'm going to have OH. Now this becomes carboxylic acid. This is aldehyde. This becomes carboxylic acid. Now we complete this one too. Now this is benzaldehyde. Look at it. This is benzaldehyde. This benzaldehyde now, what happens there? Benzaldehyde here, what are we going to do? Now, again, the same thing here. We're going to be adding OH here. This oxygen will go into this place. So we're going to draw this. Redraw it exactly the way it is. I'm going to use the shortcut. And I'm going to do this, and I'm going to put OH. So it's going from benzaldehyde to benzoic acid. You do not need to name it. Remember, when I'm testing you or assessing you on a reaction, I do not mix it up with the nomenclature. So you do not need to name them. All right, we go to the next one. We have another. Now, if you look at this question has a lot of functional groups. So the first thing it says, complete the oxidation reactions below. So you look at it, you have to find the functional groups there and know the type of oxidation reaction that will take place. Let's start. Let's look at it initially. If you look at it, this is a carbon, this is a carbon here, of course. This should be CH3. So what that tells you that this is what? A ketone. This is a ketone. That's exactly what you have at this point. This is a ketone reaction. A ketone. Then we go here. This is carbon, carbon. This is hydrogen. Now what would this be? This is going to be an aldehyde. We have an aldehyde at this point. Okay, let me try to. Yeah, okay, I can use an arrow. Aldehyde. And then what about this? You see, this is not an aldehyde. This is not a ketone. What type of this is this? This is what? An alcohol. This is a carbon. What type of alcohol? It has only one hydrogen. So it's going to be a secondary alcohol. Remember the issues of alcohol. Remember what happens to alcohol. Alcohols also undergo oxidation reaction. Primary alcohols primary alcohols undergo oxidation first to aldehyde, and from aldehyde they become carboxylic acid. Do not forget that. Whereas secondary alcohol just undergo oxidation to ketones, and they do not undergo any further reactions. Don't forget this, we did this already. So now I can mix some of these things for you in exam. So you have to be fast in thinking and remember which particular one is involved and what type of reaction will happen. And you can only do this by trying to practice as much as possible with the worksheet I provided. Okay, let's see what happens here now. So the first thing that happens here is that we know that ketone will not undergo any reaction. So if I may put mark, this is not going to undergo. This can undergo oxidation here to ketone. This one will undergo oxidation to carboxylic acid. So let's try to redraw all we have there. So if I redraw this first cyclohexane ring I have here, it doesn't look good. I'm going to be having something like this. And then, of course, this will remain that way because no reaction will take place here. Now, what about here? There will be a reaction here because this one, the carbon will still be here, but this guy will not have an OH converted to carboxylic acid. What about this one? Remember what I told you. Just remember, this is a secondary alcohol. Secondary alcohols will undergo secondary alcohols will undergo what? Reaction. Will undergo oxidation reaction to what? Ketones. So that's exactly what will happen. So we're going to put this here. So this becomes the completed reaction. Okay, we now go to the next problem. Now, this is not necessarily a problem. This here, we're talking about the application of the oxidation reactions of aldehyde. Now, this is a glucose molecule. Glucose is usually defined, or most carbohydrates defined as polyhydroxy aldehydes or ketones. So glucose is an aldehyde. How do we know that? It has the aldehyde functional group at this point. Now, the glucose-Benedict test or Benedict's reaction is an oxidation reaction. And this oxidation reaction is actually used to test for reducing sugar. It is used to quantitatively test for reducing sugar, because reducing sugars have this aldehyde, open aldehyde group that undergoes oxidation to carboxylic acid. So this is aldehyde that is being oxidized to carboxylic acid. Just the application of what we did in the previous slide. Okay, I can write that not clear enough. This is carboxylic acid. Now that's exactly, now other places no reaction occurs there. No reaction occurs in other place. The point of reaction is just at this point that this aldehyde is converted to. So what is happening there? The Benedict's test is composed of usually the copper sulfate reaction, copper 2 sulfate. So copper 2 sulfate here, usually copper has an oxidation number of 2+. Now what happens here? This is the copper sulfate. Usually it is done in an alkaline medium. So sodium hydroxide is usually used. So it's usually an alkaline medium that facilitates this reaction. So what happens here is that this skin comes in a very, very bluish color, which is the Benedict's test. Now what happens is that this guy becomes oxidized by accepting one of this oxygen from here to become carboxylic acid, whereas the copper 2 ion here is reduced from copper 2 ion to copper 1 ion, and this copper 1 ion usually formed is copper 1 oxide, and this copper 1 oxide has a brick red color, usually forms a brick red precipitate. So that's why when you do this in the lab, you find out that this reaction, when any reducing sugar, either glucose, fructose or galactose, is present, the Benedict solution is changed to brick red precipitate, telling us that there is a reducing sugar. So this is the application of the oxidation of aldehyde, and we're going to be seeing this in the laboratory as well. So glucose here is converted to an acid, and that acid is actually called the gluconic acid. Alright, we now go to the second reaction, which is reduction. Reduction, like we said earlier in the lecture so far, I'm going to say it again. Reduction is the what? Is the addition of hydrogen or the removal of oxygen. Any of them will give you the definition. So aldehydes and ketones, in this case, remember when we talked about oxidation, ketones do not undergo oxidation. But here now, both aldehydes and ketones undergo what? They undergo reduction. So aldehydes and ketones undergo reduction to be converted to alcohols. And remember again, what is just the addition of hydrogen that is taking place here. In the presence of metal catalysts, usually the two common metal catalysts used is the nickel and the palladium are commonly used catalysts. Now, what is happening here? Let's see what is happening. See, this is an aldehyde. An aldehyde is converted to a primary alcohol. Remember, a primary alcohol has either 2 hydrogens or 1 aryl group. So this is a primary alcohol. So aldehydes are converted to primary alcohols, and it makes sense because the moment this is usually 2 hydrogen, the moment you take 1 hydrogen, add at this point, and take another one and add at this point, you now have it on both sides here. So this becomes what? You now have an alcohol. So this is a primary alcohol. Now ketones, on the other hand, will accept those 2 hydrogen too. One comes here and another one goes here to form what? To form secondary. So secondary alcohol will have 2 aryl groups or only one hydrogen. So this is what? A secondary alcohol. So again, I repeat myself. Aldehydes undergo reduction to form primary alcohols, whereas ketones undergo reduction to form secondary alcohols. Let's take some reflection questions to drive this home. It says complete the reduction reactions below. We should complete the reduction reactions below. So now what do we do? Again, you have to know this. We have to know what is which. Now this is an aldehyde. What it means, an aldehyde is going to form what? An aldehyde. An aldehyde is going to form primary alcohol. Whereas this is what? Carbon. This carbonyl carbon is in between two carbon. So this is going to be, this is a ketone. It's going to form secondary alcohol. So what do? We do it immediately. So what? Remember, usually two hydrogen here. One of them is going to come and attach here. The other one is going to come and attach here. So if I repeat exactly what I have here and put this and put this, if I put this here, it's not going to convert this to a single bond of the OH, and then the other hydrogen is not going to be at this point. So this, of course, you remember there's a hydrogen here. There are two hydrogen to this carbon. That is what makes this what? A primary alcohol. On the other hand, if we get to this one, let's see what happens again. We put this, and then we have this, At this point, remember, one of these goes here, and the other one goes here. Now, we now have OH here. When this adds, it forms OH. And then, the other one, hydrogen, now comes here and adds here. Now, you see, this carbon only has one hydrogen, or two other groups on both sides. This becomes what? A secondary alcohol. So it's important you understand how this reaction takes place. You can always pause this video, try to do this yourself, and come back to see my answers and confirm that you got them. All right, we go to the next reflection question. Now, in this reflection question, it says complete the reduction reaction below. Now, again, remember, anytime you see reduction, you're going to always see me use hydrogen. Oxidation will always be O. Don't forget that. I said this by the time we started talking about this class in the review module. So now let's see what happens here. The first thing you need to do is to identify what type of functional group we have. Now, of course, this is C here. What it tells us that this is what? A ketone here. Now this is what? An aldehyde. And again, this is a secondary alcohol. Remember that. A secondary, what happens to this is reduction. Now, secondary alcohol. Know this. Alcohol is already a reduced form. It doesn't undergo further reduction. So there's no reduction. This cannot be reduced any further. Because there's already hydrogen there. You can't be adding a hydrogen to a hydrogen again. So nothing to add. So this, no reaction will take place here. So it is just there for you to think a little bit. Just a brain teaser. So the reactions are going to take place in ketone and this. Remember, ketone will be converted to what? A secondary alcohol. Aldehyde will be converted to what? This aldehyde will be converted to a primary alcohol. So again, let me draw the ring there. If I draw this ring and start with this one. Now remember, by the time there's two. By the time you add one here, add another one here. So if you add one here, this becomes OH. Add another one here, you have a secondary alcohol carbon with the OH. So that is how you know. So we go to this one. This one now, remember, if you add one hydrogen here, it becomes O. There's already hydrogen here already, but you're going to add another one here. So this has two hydrogen or one carbon alone. This carbon has two hydrogen or one carbon to this side. So this is what? A primary. Okay, I'm going to label them. Let me just label them with a different color. So this is what? Secondary alcohol. This now becomes a primary alcohol. Now what about the last one here? Now this is already reduced. No further reduction can occur. That's why we never talked about the reduction of alcohols, because they're already in the reduced state. Alright, they can only undergo oxidation, they cannot undergo reduction, just the alcohols themselves. Alright, let's move to the next thing we need to talk about. Now, what is then, like we did when we talked about oxidation, we looked at the application in the Benedict's test for identification of reducing sugars. What about this one? Now, the important application of reduction is in the biological system, in biochemistry, in intermediary metabolism. Now, reducing agent, there are a lot of strong reducing agents, just like NABH4 or NADH. NADH is actually nicotinamide adenine dinucleotide. NADH is a coenzyme. We're going to talk about this much later. It's a coenzyme. And what this coenzyme does is a strong reducing agent. What it does is to take this hydrogen and give it to another molecule. And the addition of hydrogen to another molecule becomes reduction. That's what we call it a strong reducing agent. So reducing agents such as this one and this guy deliver hydrogen ion to carbonyl carbon of aldehyde or ketones. So what is happening here is that this was happening in what we call lactic acid fermentation. You know, after glycolysis, when the pyruvate is the last reaction of glycolysis. Now pyruvate has two fates, or three fates rather. It can be broken down in lactic acid fermentation to form lactic acid. In alcohol fermentation in microbes, it can be broken down to alcohol. Whereas in aerobic respiration in tricarboxylic acid, it is further broken down to generate energy and giving out water and carbon dioxide. So this is what is happening, particularly in the muscle. This reaction is taking place in the muscle. That is where this reaction is taking place in the muscle. So this takes place in the muscle, particularly in actively metabolizing muscle, in the muscle. So what is happening? Let's see what's happening there. Like we said, it's an application of reduction reaction. Now, the reducing agent here is nicotinamide adenine dinucleotide. You don't need to memorize this name, NADH. Now, the NADH has the ability to donate two hydrogen ions, or two hydrogen ion. So it donates this guy and this guy. Look at what happened. One of them goes here and the other one goes here. And what happens here? If one goes here, it becomes OH. The other one goes here. So what happens is that it forms lactate. So this is pyruvate is now converted to lactate, and the reducing agent is NADH. So this becomes, so the NADH itself is now oxidized to NAD+, whereas pyruvate itself is now reduced to lactate. So this is an important biological application of reduction of aldehydes and ketones. Particularly, this is an aldehyde in this case because this is just the carbon and carbon between two carbon. Alright, we now go to the last thing we're going to be talking about in this video is just the reaction map for aldehydes and ketone. In this reaction map, we've discussed the phase 2 reaction, which is what? Oxidation. Look at this is oxidation. We just discussed it. And addition of hydrogen is reduction. In the second part of this lecture, like I said, we're going to be talking about the addition of alcohol. This addition of alcohol forms compound we call the hemiacetals or acetals or hemiketos or ketos. So let's start from what we know first. Now, addition of oxygen is oxidation. Oxidation, if it's an aldehyde, it forms carboxylic acid. If it's a ketone, no reaction. Remember what I told you? Ketones do not undergo oxidation reaction. Now, addition of hydrogen to aldehydes or ketone, which is reduction. Both of them undergo reduction. Now, if it is an aldehyde, it forms primary alcohol. If it is a ketone, it forms secondary alcohol, and that is it. And then if we add alcohol, which we're going to see in the second lecture, if you add alcohol, it forms what we call the hemi compound. The hemi compounds are two types. Either the hemiacetal, if it is an aldehyde, it's going to form hemiacetal. If you add more alcohol, like 2 molecules, if you add 1 molecule, it forms hemiacetal. If you add another molecule, it forms acetal. Hemi means this is the full acetal, this is the half acetal. We're going to see what this means. The same thing happens here. If you add the first alcohol, it's going to form a hemiketal. Addition of another alcohol will form the full ketal here. And it is just this we're going to be talking about in the second part of the reactions of. So the second part of this lecture, we'll be talking about just the hemi reactions and the application to the structure of biologically important molecule like carbohydrate, or the simple sugars rather. We're going to see that as we move on. And having said that, we've come to the end of this lecture. Thank you for listening. Bye at this point.